I am so grateful to welcome tonight Dr. Madelyn Espinosa-Cotton. Madelyn is a researcher, a PhD research scientist at Memorial Sloan-Kettering, and I'm so excited that she is here tonight to talk to us about her experiences and what led her to having a special interest in DSRCT. Madelyn, thank you so much for being with us.
Thanks for inviting me.
So just to get started, I am going to fix the screen. There we go. And I would love for you to just introduce yourself to the group and talk about your work at MSK and your role.
Hi, I'm Madelyn. Like Laura said, I'm a senior research scientist at Memorial Sloan-Kettering. So that means that I have a PhD, not a medical degree, but a PhD in cancer biology, basically. I went to graduate school, and then I came to MSK for my postdoctoral fellowship, and then I stayed for this role as like a staff scientist in a lab. So the lab that I'm in is in the Department of Pediatrics. The Department of Pediatrics at MSK treats most of our sarcoma patients, and a lot of the clinicians that treat patients with DSRCT they're like within our group. So I work in a lab that develops antibody-based therapies, basically. So a couple different kinds of antibodies. The first one are like bispecific T-cell engaging antibodies. So the purpose of those antibodies is to bind to something on a tumor cell that's not expressed on normal cells, so we can target the tumors specifically, and then also to draw in T cells, which are immune cells that can kill the tumors. So it serves as like a bridge between the tumor and the T cell, and it kind of shows the T cell like, hey, there's this thing here that you need to come kill. Otherwise, your T cells don't know. And then the other thing that we work on are antibodies for radioimmunotherapy, and this is kind of where most of my time is spent right now. And the radioimmunotherapy antibodies are similar to the T-cell antibodies in that they serve as a bridge between the tumor cell and this thing that's going to kill it. But instead of a T-cell, it's a radioactive metal or molecule that we can give as like liquid radiation. So we give the antibody first, we let it pre-target the tumor and kind of coat the tumor and clear away from the rest of the body. And then we give the radioisotope, and it goes and it finds that antibody on the tumor and binds to it. And so we get really specific killing of tumor cells without harming normal cells in the rest of the body. And that is all preclinical work right now. So that's not in human clinical trials yet, but I think we're really close. So most of what that looks like right now is like mouse studies. We have these special mice that we can use to grow little human tumors, and then we test these therapies and we see if they work and if they cause any kind of toxicity. And that's pretty much what I do in the lab. And then another little part is just sequencing tumors and like weird little nerdy science quests. Like the DSRCT research community is really small, so people will come to us from outside MSK who are interested in this tumor but don't have access to like tumor samples that patients have donated, but we have a whole bunch of those, so we can collaborate with them and share with them. So that's like another part of what I do.